Size-exclusion chromatography separates molecules purely based on how large they are, as they move through a column packed with porous beads. When a mixture is loaded onto the column, the larger molecules can't fit into most of the pores, so they take a more direct path and elute first. Smaller molecules wander in and out of the pores, which slows them down, so they elute later. Because the separation doesn't rely on the sample sticking to the resin, size-exclusion chromatography is considered a very gentle technique. Proteins stay folded and active, and there isn't much risk of damaging sensitive complexes. This makes size-exclusion chromatography especially useful when you want to check whether a protein is aggregated, figure out its oligomeric state, or clean up the final sample without altering its structure. It has also opened the door to studying quaternary structure and how proteins assemble and function together. Gel filtration is considered more foundational than the other methods, and is often used to complement them or polish their products. The other methods of chromatography that I will discuss later in the video are quite specific and can isolate many clinically relevant proteins like enzymes and monoclonal antibodies, but size is not so specific a metric, as many biomolecules have the same or similar hydrodynamic radius. As a result, size-exclusion chromatography's impact on human disease treatments tends to come from quality control and characterization, rather than being the primary method of purification for a breakthrough treatment. Even so, size-based separation is uniquely valuable for vaccines, virus-like particles, nanoparticles, and complex protein assemblies. One example of a clinical application of SEC is safety and proper assembly of virus-like particle vaccines, like the HPV and hepatitis B vaccines, which rely on protein shells that look like real viruses in form and size, but don't contain viral genetic material, so they are incapable of infecting host cells. These particles need to form the right size and shape without clumping together. Size-exclusion chromatography is routinely used to double-check that the particles are assembled correctly and are free of large aggregates. This figure demonstrates how a VLP-based vaccine triggers an adaptive immune response. The vaccine is first taken up by a dendritic cell, then presented by MHC1 and MHC2, which allows T cells to detect them using T cell receptors. B cells then interact with CD4 plus T helper cells, which allows plasma cells to secrete sufficient immunoglobulin G antibodies and also generate B memory cells, which essentially remember the virus for future immune responses. Virus-like particles, as purified by size exclusion, are especially useful in vaccines for several reasons. One is that they are much safer than live attenuated viruses, which means weakened living viruses, and inactivated whole virus vaccines, which contain the viral genome and could potentially replicate or revert to a virulent form. Another reason they are useful is that they trigger strong immune responses due to their highly ordered and repetitive array. This surface geometry helps cross-link B cell receptors, which causes strong antibody responses. Their size range of 20 to 200 nanometers also allows them to drain into lymph nodes and be phagocytosed by antigen-presenting cells to help activate T helper cells and B cells. Lastly, virus-like particles are very flexible. They can be produced in a variety of systems, including yeast, insect cells, plants, and mammal cells. There are also chimeric virus-like particles with foreign epitopes grafted on, such as antigens from other pathogens or from tumors. Circling back to the uses of size-exclusion chromatography for vaccine manufacture, lipid nanoparticles used in mRNA vaccines such as COVID-19 vaccines need to have tightly controlled size distributions. Size-exclusion chromatography is used in such cases to confirm that nanoparticles are the correct size, as well as identify any free RNA, empty particles, or oversized aggregates. Size-exclusion chromatography has also been used to determine if disease-related proteins form dimers, oligomers, or abnormal aggregates. Examples include the characterization of amyloid precursor protein oligomers, insulin hexamers, and immunoglobulin complexes.